You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you. Okay, you're about to hear my interview with Chamindie Punyadeera, so I hope it works. Chamindie was very nice, very accommodating. Again, as I mentioned, she just came back from traveling, was jet-lagged, but still took the time to speak to me. Chamindie is focused on several different types of cancer, neck and head cancers, glioblastoma, and one other kind. So we talked about how she wants to use what's called a liquid biopsy, looking at your saliva or your blood and finding out if you're about to have cancer, if you have cancer, how advanced it is, etc. Why is liquid so much better than a regular biopsy? Because who wants to have a needle jabbed into their neck or, a piece of their pancreas taken out or their stomach or whatever it is? I would much rather have my saliva sampled or my blood. And according to what Chamindie says, it's going to give a lot more information to do these liquid biopsies. So it's a huge, very promising area of medicine. Unfortunately, if you know someone who has cancer, God forbid, if you have cancer yourself, as I've had, if you're facing cancer or if you know someone, and unfortunately chances are many listeners do because it's so prevalent, you'll want to pay attention to this podcast because it reveals what's coming in the next 5, 10, 15 years, hopefully less than 15, about new ways to biopsy and find out what's going on with us and how we can be helped with medicine. So definitely listen in.
Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast. I have a guest from far away in Australia, Chamindie Punyadeera. She got up really early to do this podcast, and I appreciate it. I have the luxury of doing this in the afternoon. I'm wide awake, and Chamindie got up super early after just traveling. So I'll try to be nice to her. But Chamindie, thanks for coming. How are you doing?
Good morning. Hi. I'm all right. Thank you.
Oh, good. Well, tell me about your work. I know that you're working on cancer biology, and it looks like specifically possibly head and neck cancers or maybe other disorders. Tell me about your work. What do you research?
Before I start talking about the research that we are doing in my team, I would like to start off by saying why it is important. Why is it important to do the research that we are doing? So healthcare costs are increasing globally due to a growing and an aging population. We are trying to develop non-invasive diagnostic tools to detect diseases early, in particular cardiovascular disease and cancer. The 3 types of cancer that we are researching currently are on head and neck cancers, lung cancer, and glioblastoma. We are using human saliva as a medium to identify disease-specific biomarkers in saliva. You may be wondering why saliva. Saliva collection is easy, non-invasive, and multiple samples can be collected by an individual. And it's also ideal for rural and remote setting. In addition to salivary diagnostic, we are also using liquid biopsy to diagnose and predict treatment responses in cancer patients. Liquid biopsy, by definition, is the use of body fluids, such as blood and saliva, in place of tumor tissues to manage cancer patients. The rationale for using liquid biopsies are as follows.
Well, I think I know the irrationality is that I've had thyroid cancer myself, and I had a biopsy of nodules in my neck, and it's no fun to have a needle plunged into you and stuff aspirated out. So I would have loved to have instead just give a saliva sample or a blood sample. It's a lot less invasive, and I'm sure for a lot of cancers it's far more invasive than what I went through. So I think that would be a big reason why it would be nice.
Yes, and I'm so sorry that you had thyroid cancers, but thyroid cancer prognosis is about more than 90%. So it's kind of a good type of cancer to have compared to head and neck cancers, where survival is 50%.
Yeah, actually, I'm glad it happened because it changed my perspective on things. I feel lucky because I guess, knock wood, at least so far, everything looks good. And if you're going to get cancer, it's probably one of the best ones you can get, which is a weird thing to say. So.
Absolutely. Let me just go back and touch upon why liquid biopsies in place of traditional tumor tissue, because tissue biopsies are invasive and some locations are difficult to access, such as when you get tumors in the lung and the brain. Secondly, single biopsy can miss relevant tumor clones due to intra-patient tumor heterogeneity. And lastly, sequential tissue biopsies in individual patients for real-time monitoring of therapy responses are less feasible actually in a clinical practice setting. So these
Let me ask you a quick question here. You mentioned that tumors are heterogeneous, meaning they're not all one thing, one cell expression type. So yeah, you're right. If you do a biopsy, there's no guarantee that you'll get enough of the heterogeneity to really understand the tumor, I guess, right?
Absolutely. Let's just take your example for a second. For thyroid cancers, you said you did a fine needle biopsy. How do we know that you actually get the tumor cells? Because the tumor itself is quite heterogeneous. You need to be doing multiple biopsies to be able to get a actual picture of the tumor tissue itself. But in liquid biopsies, when you are using circulating tumor cells, for instance, these are the cells that are disseminated from both primary and metastatic sites, and they actually represent the actual clone that's giving rise to secondary mets.
I guess the trade-off is, you may be getting differing signals from, 20 different, I don't know if you want to call them cell types, but in a tumor, what do you call the different cells that make up the heterogeneous tumor? They have different epigenetics, different gene expression, but what do you call them? Are they called cell types, or what are they called?
So it's both cell types, so different cell types, because it's a tumor microenvironment, which means the interactions of tumor cells or epithelial cells with immune cells, stroma cells, that whole interaction is what leads to cancer. So by sampling one cell types, you would not get the actual expression of different markers in the whole tumor tissue. So using liquid biopsies, you do, because it's the clone that can resist in blood, gives rise to secondary mets. So you know that clone will give a secondary mets, and you are able to isolate and characterize these cells from a cancer patient's blood sample.
Okay, so whatever biomarkers you're looking at, you can look at the relative prevalence of the different markers that could come from tumors and metastatic sites. Okay, I see what you mean. So, but what are the biomarkers you'd look at in a liquid biopsy? What are examples of some of them?
So as I said, liquid biopsy by definition is using liquid such as saliva or blood in place of tumor tissues. So in my team, we have developed a workflow to isolate and enrich circulating tumor cells from cancer patients' blood. This is really important because circulating tumor cells, or in short CTCs, these are rare cells. These are about 1 to 5 cells per 7 mils of blood sample. So, it's like finding a needle in a haystack. And it's really important, the first step, the enrichment step, because you may miss CTCs because these are very, very rare events. But with our workflow, we are quite confident that we can isolate, detect these CTC. And secondly, we are also using cell-free DNA. So this is the DNA that's secreted into the blood or saliva through apoptotic events, necrosis. And we are looking at the genetic profile of these cell-free DNA, which is called then ctDNA. And what we do in head and neck cancers is that we are monitoring minimal residual disease using ctDNA. Currently, there are FDA-approved platform for using ctDNA in non-small cell lung cancer and breast cancer. We are also using exosomes. These are extracellular vesicles that are secreted into either blood or saliva. And these are called the cargos. These are the vesicles that communicate between different cell types. So a cancer cell can direct a normal cell to become cancer cell. So these exosomes are really important both biologically and clinically. So these are the type of biomarkers.
Question here. Yeah. So you were saying that cancer cells are very few in the blood, for instance, or in the saliva. What about exosomes from cancer cells? Are those a lot more prevalent? And can you even identify, oh yeah, these exosomes are from cancer cells? Because I would think there's exosomes from all kinds of cells in the blood or saliva. How would you ever tease them out?
Absolutely spot on. So exosome biogenesis or exosome secretion is a physiological event. So even in healthy cells, normal cells, they secrete exosomes. And the exosome cargo content secreted from a cancer cell differs from a normal healthy cells. Let's take, for example, breast cancer. You could detect HER2 receptors in exosomes secreted from a breast cancer patient compared to healthy cells. So we are actually looking at the cargo in extravesicles, not per se the extravesicles, because these molecules are the same, but the cargo differs between normal and healthy cells. Normal and healthy cells.
Even though even though there's many different types of exosomes, you can identify ones associated with cancer or, from cancer cells. How numerous are the exosomes? Do you have any idea? And is it, if they're very numerous, does that make it easier to gauge their signal and see them versus the tumor cells themselves that are in blood?
So we are just now finalizing a research article on isolation and characterization of exosomes from head and neck cancer patients using saliva as a diagnostic fluid. So we do get a large number of, I think, 10 to the 6 particles in saliva of cancer patients. And more so, we are using a battery of markers to characterize exosomes. We are using CD9, 63, 81 as markers, as well as we are using cryo-SCMs, because exosomes are between 50 nanometers to about 150 nanometers. So when you isolate these exosomes, you have to really characterize them to show that these are exosomes and not other extracellular vesicles such as apoptotic bodies or multivesicular bodies.
So in order to see what the cargo is for exosomes, do you have to ultracentrifuge a sample down, and would that break open the membranes of the exosomes and spill their contents, and you look for specific RNA signatures inside them, or can you preserve the exosomes and look at them that way?
So we are using, there are a large number of exosome isolation protocols, but we are using ultracentrifugation, and we can also freeze these exosomes, and we've never had a problem of exosome bursting or things like that. So yes, you can store exosomes, and then you can do gene expression, microRNA expression to identify tumor specific signatures in these exosomes. And what you normally do is you isolate exosomes from, let's say, a cancer patient, in this case, head and neck cancer patient saliva, and you have a healthy control group where you also isolate exosomes, and then you compare the cargo between the two groups.
So are you trying to find early incidence of disease, or are you trying to find, how advanced a particular cancer is? Like what's more of the goal here, or is it both?
So for us, we are using it to early detect cancer events, in fact, in pre-malignant stage, as well as to stratify patients at diagnosis to see whether a patient is in an early stage versus a late stage of cancer.
How do you stratify? Does the nature of the exosomes or the nature of the tumor cells that are in the blood or saliva, does that change? Does a primary tumor establish first and then grow to a certain size and then metastasize, and maybe the nature of the exosomes put out changes and the cells put out changes?
Yeah. So let's take throat cancer, which is called also oropharyngeal cancer, which is caused by human papilloma virus. So the two high-risk groups are 16 and 18. And we've collected and characterized HPV 16 DNA in the exosomes of these cancer patients. So you get a large number of, if a patient is from an advanced stage versus an early stage, which is, I think, pretty obvious, because early stage cancers, you have small tumors compared to late stage cancers or metastatic cancers. So in that way, we can see whether a patient is from an early stage versus late stage cancer.
Okay, so the number increases dramatically, I guess, because the tumor is in constant growth mode. So as the tumor grows, more and more of its cells are pumping out exosomes saying, let's spread, or communicating whatever they're communicating. We don't know, but that's why the correlation.
And it's really a fascinating field, because just take, now I'm going to deviate a little bit from cancer into cardiovascular disease or heart failure, because that's a systemic event. We can detect cardiac specific markers such as cardiac troponin I in saliva. And I have not been able to prove it, but I think exosomes are the reasons why we can detect these heart-specific markers in saliva. So literally exosomes link between saliva and the blood, and we can use saliva as a proxy to detect systemic events.
Are you, I wonder, well, this may be off topic, but I wonder if people are able to determine large sinks for exosomes. Let's say they go into the blood and the saliva, but are there any areas where the exosomes really tend to fall off quickly? Let's say you do a blood sample. If you do it from the arm, do you think you'd see a different profile of exosomes from the leg, or from if you took blood from like a central line?
That's an interesting, very interesting research questions. I am not sure whether that has been looked at. Our focus thus far has been on saliva. So we literally take about 0.5 ml of saliva, and we isolate exosomes from saliva, and we hope that it's more homogeneous. But I can answer that with the circulating tumor cells. Studies have shown that if you use right arm versus left arm, the number of CTCs you get are different. So let's say left arm gives you 3 CTCs, right arm gives you 5 CTCs.
Okay. Well, since you're sampling saliva, I know that, and this will go into another area, there's different microbial constituents based on, any fluid you sample, blood, saliva, etc. So if you're going to sample saliva, have you tried sampling from different areas in the mouth, that have different microbial constituents, and maybe that changes what you're able to see? I don't know. Maybe not. That might be something to look at.
So there are 3 main methods of sampling saliva. One is called unstimulated saliva. Like as I'm sitting, you can tilt your head down and you can pull saliva in a resting mode. That is unstimulated saliva. But sadly, most of our cancer patients, post-treatment, they have impaired salivary glands. So they are unable to secrete sufficient saliva. In that way, we give them a lemon lolly. So that's called acid-stimulated saliva. or we can give them something to chew, and that is called mechanically stimulated saliva. Concentration of biomolecules are more in unstimulated saliva versus stimulated saliva. And because we are using whole mouth saliva, which is secreted from the 3 major glands plus the minor glands, it kind of represents the whole oral cavity. And we are not particularly using or swabbing just on the tongue or at the back of the mouth, but the whole mouth saliva. So let's take the microbial compositions. They do differ. There have been studies where they have sampled different parts of the oral cavity and they find different bugs, I wouldn't call different microbials living in the oral cavity. But we are targeting human-specific gene expressions and not microbial-specific gene expression. So the primers that we design are human-specific. So you really are not amplifying the microbial communities, but you are looking at the expression changes in human cells, if that makes sense.
Okay, yeah, I gotcha. So interesting. What in terms of I didn't ask you about the heart risk factors. What can you see by looking at saliva about the heart and the cardiovascular system? Like, what kind of things can you look for? What kind of markers? And what does that tell you?
Yeah, so we are working on heart failure, because it's heart failure by definition is the inability of the heart to pump sufficient blood to the body. And there are over 38 million people globally diagnosed with heart failure. And sadly, 50% of them die within 5 years. So when we started this program about 10 years ago, what we started off with, the premise was that can we detect current diagnostic biomarkers such as NT-proBNP in saliva of heart failure patients? And yes, we can detect NT-proBNP, which is a current blood-based diagnostic biomarker in saliva, but the concentration ranges are low in saliva. In saliva, you get them in picogram per mil ranges, and as such, you need highly sensitive immunoassays to be able to pick the low concentrations in saliva. I did mention about the cardiac troponin I, and we also saw a correlation between cardiac troponin I levels in blood and saliva, which indicates that what is in the blood is what's getting filtered into saliva. So we could use cardiac troponin I as a marker in saliva. And in addition to that, we've also done a discovery approach where we have identified, where we have used mass spectrometry to identify and validate a panel of protein to detect heart failure, which we have filed a patent and which has now been licensed to an Australian-based startup company.
Okay, interesting. Well, why do you think that you're able to see markers in blood and saliva? But is it that the heart is in a stress state and therefore it's communicating what's going on with it? Or is the heart always communicating through the use of, let's say, exosomes about its state?
So if you take salivary glands, these have to be functional. So they have capillary networks. And studies have shown that biomolecules can move from blood into saliva by diffusion, active transport. There are different mechanisms how molecules get transported across the blood endothelium into the salivary acini cell. These are the salivary gland cells. So there's continuous flux or continuous traffic between blood and saliva. And these molecules continuously move either side. And we can actually capture these transportation of these molecules at a certain time when you sample saliva. And I'll give you an example. So let's go back to our oropharyngeal cancer case, and we have been able to detect HPV-16 DNA in salivary oral rinse samples from cancer patients. And when you remove the tumor, salivary oral HPV DNA content is decreased, which means that this is a good marker to diagnose and predict a treatment response in oropharyngeal cancer patients.
Okay, that makes sense. What other nuances, so we all want early detection, which would be great, but what about the overall functioning of someone's cardiovascular system? Are you looking at all at healthy people, maybe looking at them over time or under different conditions, stress conditions, and seeing what shows up in the saliva or blood? Again, how much broadcasting of the state of the system of the body, again, what the heart's doing or what the cardiovascular system's doing is happening on a regular basis. Have you looked at that baseline?
So we have actually collected about 800 samples from as part of one of the largest longitudinal clinical trials conducted in Australia, which is called Screen Heart Failure Program, where where Monash has been very kind enough to collect saliva samples from high-risk individuals. So these are people at risk of developing heart failure. These could be people with type 2 diabetes, obesity, people who have previously had heart attacks. And what we now want to do is to compare the saliva markers or proteins in this high-risk group to control group and see whether we can discriminate these two groups. If we can discriminate these two groups with a high sensitivity and specificity, we have a marker to start screening within a population. But we have collected the samples, but sadly I haven't been able to get research funding. So I have kind of parked that project until I get funding to start looking at the expression changes between these two cohorts of people.
But I guess the good thing you mentioned is that if it's exosomes, they're stable and they can be frozen. So perhaps long-term, they can be stored, and then when you're ready, look at them.
Absolutely. And even saliva, we've collected, the way we process and store saliva, we can keep salivary protein without being degraded for a longer time.
Oh, interesting. Okay. Well, excellent. So just to state it very clearly, what do you think will be the outcomes for human health, over the next few years because of your research? What's your goals there?
So I think what we want to drive is a precision medicine approach. So in other words, giving the right patient the right therapy, the right time. So if you do recall 2015 President Obama's Moonshot Initiative, which means that identifying the genetic profile of a patient and providing the treatment to that patient cohort, which is precision medicine or personalized medicine. And liquid biopsies will be the answer to accelerate precision medicine. I mean, we all know that one glove fits all doesn't work in clinical practice anymore. So it's the evolution of precision medicine approaches using liquid biopsy and tailoring a cancer treatment for a particular patient group. Just take an example of the poster child of personalized medicine is Herceptin, drug Herceptin, which is given to 20% of breast cancer patients who have overexpression of HER2. So something like that would be possible. I am very confident for head and neck, and in lung cancer, they are already doing that type of work where patients with, let's say, EGFR mutations, they get tyrosine kinase inhibitors. So you can stratify patient groups and deliver treatment based on their genetic profile. This will be the future.
Interesting. Okay. Yeah. Well, that makes a lot of sense. Have you gotten to a point where you could look at the, what's being expressed, the biomarkers across patients at a similar stage with the same type of cancer or the apparent same type of cancer to see the variation?
Yeah. Yes. So we've been very fortunate last year and I have to acknowledge the funding body, Cancer Australia, have been very generous, and they have given us research funding to conduct a longitudinal trial. And this longitudinal trial, what we are aiming is to see whether we can use circulating tumor cells at diagnosis as a prognostic biomarker in head and neck cancer patients. So we collect saliva and blood samples at baseline 13th week. The reason why we are collecting at 13th week is that's when they get a PET CT scan, and that's when a clinician can evaluate whether the patient has responded partially or completely, or whether the patient still has minimal residual disease. And then we collect samples every 6 months for 2-year period. The reason why we selected 2-year window is that most of the head and neck cancer patients, recurrences occur within 2 years. And we are collecting blood samples from 100 patients, and we have currently collected, at least at baseline, 59 samples with follow-up. And initial results indicate that, yes, at diagnosis, we can say which patients will develop recurrences and who won't develop recurrences. This is a huge, this will change literally the current clinical practice in managing head and neck cancer patients. So we are really thrilled with these initial findings and looking forward to completing this trial in the next few years.
Know that someone's going to have reoccurrence or not? What tells you that? What's the difference?
So if you look at the CTCs at baseline, and let's look at the CTCs at 13th week. Circulating tumor cells, if a patient is responding, should decrease. Let's say at baseline you have 10 CTCs, and post-treatment, if it decreases like into 5, 3, 2, 1, you can see that within 2 years, patient is responding, as opposed to you have 5 CTCs and the CTCs keep on increasing. Then you know that the patient is not responding. Also, another marker that's currently being used in clinical practice, like Foundation Medicine and Guardian 360, they're using ctDNA and tumor profiles, not mutation profiles, to detect minimal residual disease in lung cancer patients and breast cancer patients. And we would like to do a similar thing in head and neck cancer patients.
Okay. So, all right. So you're observing these biomarkers pre-treatment and post-treatment. Let's say you observe them pre-treatment. What if you did it over a couple of time periods and you saw that, the CTCs were increasing tremendously fast, and in some other people they were slower? And then if you also did it post-treatment and you saw they're falling off fast versus falling off slow or not at all, I mean, I would think that both sampling periods would really help you characterize what's happening with someone.
Yeah. So for in head and neck cancer patients, so if the tumor is, let's say, oropharyngeal cancer patients or throat cancer patients, they basically get chemoradiation, as opposed to oral cavity cancers or mouth cancers where they remove it surgically. If, for instance, you see that the CTCs are increasing, a clinician could intervene and do multimodality treatment, surgery plus chemoradiation or cetuximab or immune therapy. You see, you have more option to aggressively treat the patient at early onset rather than late onset, where the survival will be less than 50%.
Well, I guess, again, you could also characterize a degree of aggressiveness of their cancer if, by looking at biomarkers and seeing their rapid change versus their slow change.
Absolutely. Yes, you're right.
Okay. And again, because it's, it's not invasive and it can better characterize what's going on, it can be done more times. It probably will be a lot cheaper, and it'll lead to better care for people.
Yes. So liquid biopsies, just take, for example, a cancer patients who had surgery. How do you track this patient's disease progression or regression? Because the tumor is removed. What do you use? You use either saliva and blood biomarkers to track this patient's treatment response. Because once a tumor is removed, unless you have in an unfortunate case, you develop secondary metastasis, there is no tumor to track. But you can get the biomarker profiles to track, or biomarker tracking.
Okay, I see what you mean. Well, very good. I mean, there's a lot of promise, a lot of work to do as usual, but this is great. So, Chamindie, what's the best way for people to learn more about what you're doing and read papers and interact and ask questions, maybe?
So I would actually direct them to QUT. So I'm from Queensland University of Technology in Brisbane, Australia. And our library is running e-prints. So all our research articles are on, if you put my name, Chamindie Punyadeera, QUT e-prints, you can download all the research articles. And I do a large number of TV, media, and talks. And I'm also engaged with the community, where I do a lot of community events and explain the importance of early diagnosis of cancer and cardiovascular disease. So if you just put my name and QUT e-prints, you can get all the articles that we have published from my team.
Okay, well, that's great. Well, Chamindie, I know, again, it's early. You've had a lot of travel and all that, but I really appreciate you being here. It's been a great call, and thank you so much.
And thank you so much for doing this, because it's, as researchers, we really rely on funding. And the more we can communicate our research outcomes to the public, then we can hopefully in the future get funding to do this exciting research where we can do a paradigm shift in clinical practice.
Well, very good.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.